Hello all, my name is Ryan Abdelaziz and I am responding to the initial post for the VoiceThread 2 assignment. So for this discussion, my position is that exploratory data analysis is essential to scientific discovery, but it becomes problematic when the analytical process is hidden or when exploratory findings are presented as confirmatory evidence. So first, where is the line between legitimate exploration and data dredging? So exploratory data analysis is not simply testing different models until one produces a significant result. Its purpose is to understand the structure and quality of a dataset by examining distributions, outliers, missingness, batch effects, nonlinear relationships, and model assumptions. It can also reveal biological patterns that were not anticipated before data collection. The line is crossed when researchers use analytical flexibility selectively, testing many outcomes, subgroups, transformations, covariate sets, or exclusion rules, and reporting only the successful results is closer to p-hacking than legitimate exploration. For me, the key distinction is whether the final claim accurately reflects how the result was obtained. A pattern discovered during exploration should be labeled as exploratory rather than described as though it had been predicted in advance. A preliminary study I'm involved in, involving ADHD, GWAS, and EQTL, illustrates this point. The association between ADHD phenotype and the genetic risk measure appears modest. It would be inappropriate to conclude immediately that diagnoses are inaccurate. Several explanations remain possible, including diagnostic and phenotypic heterogeneity, environmental contributions, limited power, ancestry differences, or incomplete measurement of genetic liability. EDA can help determine whether the pattern differs across subgroups or is influenced by technical and demographic variables. But those observations would generate hypotheses rather than confirm a single explanation. This ties directly to the first question. Legitimate EDA considers multiple plausible explanations and reports them transparently. Data dredging searches across enough analysis to obtain a favorable result and then presents that result as uniquely meaningful. Second, is HARKing always a scientific failure? I do not think that developing a hypothesis after observing a result is inherently unscientific. In genomics and multiomics, datasets are too complex for every meaningful relationship to be predicted beforehand. Unexpected findings are often where discovery begins. The problem is not post hoc reasoning itself, the problem is concealing when the hypothesis was formed. HARKing becomes misleading when a researcher presents an explanation developed after seeing the data as though it had been the original hypothesis. I'm doing another GWAS study on anhedonia, and that illustrates this tension as well. So the study begins with GWAS loci associated with anhedonia and asks whether these loci regulate gene expression through RNA sequencing and EQTL analysis. Because anhedonia occurs across several psychiatric disorders, many diagnostic groups could potentially be added. That flexibility is scientifically useful, but it also increases the number of possible comparisons. A rigorous approach is to define the primary diagnostic contrasts, biological rationale, covariates, statistical model, and multiple testing strategy before the main analysis. Additional diagnostic patterns can still be examined, but they should be identified as exploratory and validated in an independent dataset or follow-up study. This answers the second question. Post hoc hypothesis generation can be reasonable and productive. It becomes a scientific failure when the timing of the hypothesis is hidden or when the same data are treated as independent confirmations of the idea they generated. Third, should EDA be formally governed? I support a hybrid approach. So, confirmatory, hypothesis, and primary analysis should be documented in advance when possible, but researchers must retain enough flexibility to respond to real features of the data. I'll use my anhedonia project again as an example. So, for my project, access to institutional data requires a clear hypothesis, justification for the requested diagnostic groups, and a proposed analysis plan. This type of pre-analysis review can improve rigor because it requires the scientific rationale and methods to be articulated before the results are examined. However, no analysis plan can anticipate every issue in a complex dataset. EDA may reveal batch effects, missingness, nonlinear relationships, outliers, or poor model fit. Researchers should be allowed to adapt, but those changes should be documented. Exploratory analysis should be distinguished from pre-specified tests, and major deviations, multiple comparisons, and sensitivity analyses should be reported. I also support open science, but open access may be balanced with privacy, must be balanced with privacy. Human genomic and clinical data may require controlled access because of re-identification risk. A reasonable model is broad access to de-identified, low-risk data, controlled access for sensitive datasets, and open sharing of code, metadata, and analysis plans whenever possible. My overall position is that EDA should be governed through transparency rather than rigid restriction. Exploration generates possibilities. Confirmation tests whether those possibilities persist under a fair and independent evaluation. Unexpected findings are not the problem. The problem begins when an unexpected result is represented as though it had always been expected. Thank you very much for listening. I look forward to seeing any thoughts or discussions you guys have on my, discussion. And yeah, look forward to hearing from you all. Thank you.